Hey, how's it going, guys? This is Paul again with sphericreptiles, and as the title says, today we'll be covering how to calculate Hey, how's it going, guys? This is Paul again with sphericreptiles, and as the title says for this YouTube video, we'll be covering how to calculate the genetic odds for your snake. In particular, we'll be talking about ball pythons, where there's a lot of different questions and there's a lot of morphs that you got to deal with. So if you're not buying a basic ball python and you're actually trying to invest in it, or you want to make sure that you're getting what you paid for, it's important for you to know how to calculate those genetic odds, so when you're talking to the breeder or the seller, that you can know exactly what you're getting. So without further ado, so we got that part, the title, and the second thing, I'm going to go over some of the descriptive terms that we'll use to kind of describe things so that you can make sense of it. Of course, DNA is the backbone of your genetic code, and we're going to call that like the house. So everybody, you know what a house is, and the chromosomes are the room. So the chromosomes exist on the DNA strands, the double helix strands, and then you have genes. So you have genes or alleles, and those genes on those alleles would represent the furniture, the paint, the textures of the room, and etc. So anything that you have in there that would tell someone else that this is your house with your rooms and your stuff in it. So that's all of you, basically. So that's what we'll use for those terms. The second thing that we'll cover are dominant genes. So dominant genes, they overshadow the alleles or the genes that are recessive. So every time you see a dominant gene, it's always expressed. Now, so you're going to have two different kinds of dominant genes. You're going to have a codominant gene and an incomplete dominant gene. Well, the best way to remember codominance is that if you combine with each parent, they're expressed. So the best way that I can say that happens is if you have, like, I think the common thing that we use, I went to UTSA, so this is very, very, this is a long time ago, 99, So if you have a black cow and a white cow, you codominant genes would be expressed in a black cow with white spots or a white cow with black spots, however you see it. So that would be codominant, where both of the parents are seen. Like it'd be almost like a spotted cat or something like that. Now, incomplete dominant is the exact opposite. Incomplete dominant, what I would like for you to remember is that it's a blending to make a new look. So the best example I can think of is if you have a red flower and a white flower, you mix them together, you blend them together, and you end up with a pink flower. So that makes a whole new look. The only codominant gene that I know would be for like scaleless pythons. So scaleless ball pythons, the entire body might not be scaleless. Only maybe the head scaleless, or certain parts of the body are scaleless. The other parts have scales, like maybe on the underside or something like that. So that would be your only codominant gene. All the other genes that are dominant in ball pythons would be incomplete dominant. So that would be anything from like yellow belly, you might have acid, you might have like pastel, you might have fire, some of those things like that. So that gives you a kind of sense of what we're dealing with. So we're going to X these out with the exception of that one type of python where there's an actual morph change where you can see both parents. And then now we're going to talk about incomplete dominant. But before we can move on, we have to discuss recessive genes. So in order for recessive genes to be expressed, you have to have a pair or a copy from the male and the female parent. So you have to have two lowercase r's or two lowercase of whatever is the way that it normally looks. And so if this is a large R, you'd have a small r. So if you have two of these, this would be your recessive. Now this would be your dominant, or this would be your dominant. You can see either way, you're going to be dominant there. So you're going to have whatever copy of that gene that's from that parent, male or female, it will override that recessive gene. Now the next thing we got to talk about, this comes into play when you're trying to buy things that aren't visual. So visual means like something that you can see. So if we have homozygous, we got two identical alleles for a gene. So if you have pastel, and pastel. You end up with a super pastel because it's an incomplete dominant. You could end up with that. Then you know that that's a homozygous. So you can have homozygous genes, you can have, but you need them to be homozygous for recessive traits, which is why you have to have two R's and not a big R and a big R. This would equal the next thing, which is heterozygous. So when you have two different alleles for a gene from either parent, in the case you have a dominant gene or a normal gene and a recessive gene, that's heterozygous. So after we move forward from that, next thing we'll discuss are genotypes and phenotypes. These are so closely related that a lot of the times people won't even discuss what the phenotype is. Now, a genotype is a genetic combination of the alleles for a specific gene. So that's whether you end up with an acid orange dream, you end up with a yellow-bellied clown, something like that. So that's the genotype. So that's basically saying that's the code for that expression of that gene, however it looks. Incomplete dominant, codominant, recessive, whatever it is. That's the genotype. Now, what you visually see or what you can put your hands on, that will be the phenotype. And so that's the physical characteristics of a genotype, and that's what you can see. So guys, just take a look. I know that you can always pause this and go back and look at it again, but hey, those are the terms that we'll use. So now we'll discuss some of the Punnett squares, as they're called. So we got dominant genes, which are all or none. So the examples I have here, and then we're talking about incomplete dominant, are acid, calico, or sugar, leopard, pinstripe, redhead, spider, stranger, woma, and Wookiee. Those are the examples that I'm just putting out. There are a lot of others, guys. I mean, if you look at the combination of ball pythons, they're almost endless. But if you look at the overall backbone of the look, most of those genes will be dominant. So in the case that I have now is if you have an acid ball python mixed with a normal, you got a big A for, but this is actually like normal. So the big A's always represent normal. And you got, so you got an acid and a normal. Now this is acid, it's just say big A, small a. So this is the acid gene and that's the normal. Now this acid, wherever you see this big A, that will be represented. So you have the normal with a not copy. So that would be with this means like it's not expressed. So you could have a big A and a big A where you would have all acid. But for this case, we're going to say it's heterozygous. So you have acids here and normals here, as you can see. So you end up with half acid, half normal. Now, the second example I have is if you have heterozygous acid with a heterozygous leopard. Now, I could have put leopard and acid, but I don't think that you would see. So that would be 100%. If you had a homozygous leopard genes and a homozygous acid gene from the parents, whether it's male or female, you'd end up with all acid leopards. But for the point of this exercise, we're going to say that they're heterozygous. So from this Punnett square, you end up with a quarter of the animals ending up acid leopards, or they have a quarter of a chance per egg. So 25% chance per egg to becoming out an acid leopard, an acid, a leopard, or a normal. So to put this in perspective, so if you had 4 eggs, you could end up with all normals. You very well could. Or you could end up with a quarter of them leopard and a quarter of them, you could end up with a straight quarters for all of them. Or you could end up with all acid leopards. So for each egg, this is the probability that you're going to get that. So you think about Plinko from The Price is Right. Every time you drop that Plinko chip in there, you could end up with one of these 4 options. So that's dominant, and now you have, acid to normal, and then you have an acid to a leopard. That's just an example I use, but you can use any example that you'd like. So now let's discuss the incomplete dominant blending. So the best thing that I can discuss with that are the blue-eyed leucistic complexes. So you have bamboo, special, Mojave, mystic, Russo, honey, phantom, the daddy gene, mocha, and lesser. And these are all, basically going to be either heterozygous, but when they're homozygous, they'll introduce themselves or they'll show a phenotype or a physical showing of a blue-eyed leucistic. That's what BEL stands for, blue-eyed leucistic. So let's look at the first example where we have a bamboo to a normal. Of course, this small b means that it's a normal. So we have the normal genes here represented by N's, and capital B's and lowercase b's, so it's a 50-50 chance as to whether you get a bamboo or a normal, and that's per egg. So that's one of the things I don't think people really realize, is that it's not like if I had 4 eggs and half of them will be bamboo and the other half will be normal. No, it's per egg, there's a 50% chance that you'll end up with a bamboo or a normal. I think that makes sense now. It's a coin toss for that. Now, if we go to the second, we'll look at pastels versus the Mojave. So it's big M, big P, small p. So we can see here from this, the way that this works out is that 25%, there's a 25% chance you'll end up with a pastel, which is a pastel Mojave, 25% Mojave, 25% pastel, and a 25% chance that you'll end up with a normal, which is this one down here, small p, small m. Of course, I'm just using the heterozygous. And now if we go to the incomplete dominant for the blue-eyed leucistic class of snakes, I'll call them, that we'll go to the third option where we have a Russo with a mystic. So what we have is big M, big R, small r for the Russo, and right here, in this top one, we have big M, small r, we got a blue-eyed leucistic. Then we have 25% chance of getting a Russo, a 25% chance of getting a mystic, and a 25% chance of getting a normal, which is down here. So again, guys, that doesn't say that every time you breed a blue-eyed leucistic that you would end up with, not every time that you breed a mystic or a Russo or any animal that's part of the blue-eyed leucistic complex, that you'll end up with a blue-eyed leucistic, all white snake with blue eyes. But there is a 25% chance that you will per egg. So your chances are pretty good if you think of 1 in 4. So out of a clutch of 6 to 8 eggs, you probably will end up with 1 or 2 of them. You might end up with 3 or 4. So, hey guys, let's review that chart. If you have any questions, please comment or ask me a question, and we'll move right along. So now we'll discuss the recessive genes, which you remember are designated with small r, and there you need to have a double recessive to be a visual or to show. So you got 2 small r's, so it's homozygous. And the first example I'll use is a clown to a clown. Of course, you can look at the picture here on the screen of the clowns that I'm going to use for this example, just to give you a visual expression. You have a clown male with a clown female. They're both double recessive or homozygous. So you got small c, small c, small c, small c, and the resulting offspring will be 100% clown for all of the offspring. Now, that's not to say if they have an incomplete dominant gene attached to them, like a pastel or a fire, then that's a different combination. But that's just, you just work that out the same way. And the second one that we have, the second example, is if we have a homozygous clown, which is small c, to a heterozygous clown, which is capital C, small c. But this capital C in this case represents normal, because you have to have homozygous expressions or a double recessive in order for it to show. So anytime you see a capital, that's a normal. But I'm just trying to tag it with that same spot on the allele or gene so that you know what I'm talking about. Whereas here you have a heterozygous male, it's a normal with a recessive trait for clown. Then you have a female with a double recessive. And what you'd end up with, as you can see, is you have a 50% chance of getting a clown visual from this side and per egg, and then you have a 50% chance of being 100% het for clown. And so that, but the hets will always have a normal appearance. That's one thing that I really want to stress. And so sometimes they'll have markers in them. They'll have ringers in the case of pied pythons. But more or less, you know that this is the best situation you could have outside of 2 double recessive parents or 2 visual parents mating. So the clowns, you know what they are double recessive. But this guarantees you that you got 100% het clown. So with this one, you'll see that a lot, that it's 100%. And that means that every time you have that trait. You have 1 recessive trait, so then you can play those odds. Now, the way those odds would work out is if you were to breed 2 heterozygous parents. So it could be pied, it could be albino, it could be clown. I know I've used clown a lot, but it could be clown or any of those other things. It could be sunset. It could be any type of double recessive visual expression of a pattern morph that you could work this Punnett square out with. But in this case, we're going to talk about clowns, so just to keep it consistent. So I got a heterozygous male and heterozygous female. So you can see where the big C's are the normals and the smaller C's are the recessive clown traits. So you do have a 25% chance of getting that normal, just all the way normal. There's no clown recessive trait in it. Now, the interesting thing is when you see this, when you see 66% het clown, what does that mean, right? So if you get this visual, then you're good. But if you do these right here, you'd have to genetic test, because there's no way for you to tell if this non-visual heterozygous clown is just a normal without genetic skin testing or breeding it out. So luckily, we do have genetic skin testing available so that when you do get one of these 66% het clown or het recessive, if you're trying to see if you hit for a het, then that's one way that you could tell right off the bat. And if someone's did that, they're probably going to charge you for the 100% het, because that's what it is. Once it tests heterozygous, and you know it's got a normal and a recessive trait. So guys, that's what we got there, but you got to remember that all of these have a normal appearance, whereas only this one has a double recessive for the clown trait. Now, the next example I have is one that you could probably gamble on, but it's a weaker bet than you would if you were to take, of course, the heterozygous to the double recessive parent. And that would be an example for the het clown. It's 100% het clown with a normal ball python. So sometimes that's how you can kickstart it if you don't have the cash to do it. But this is how that would play out. So if we said that the male was heterozygous and the female was normal, so I have the big C and the big C, those equal normal. Then, so you would see that you have a 50% chance of just having a normal, or you have a 50% chance of having a heterozygous. Visually or phenotypically, there's no way for you to be able to tell the difference between these two. There's no way without genetic skin testing. So that means that half of the snakes will be normal, and then the other half will have het clown. But without genetic skin testing, this is the lowest chance of you hitting what you want. So if you see this 50% het clown or 50% het, whatever recessive trait you're trying to get, then you notice that the prices are a lot lower. And the reason the prices are a lot lower is because there's no guarantee. Now, if the seller or the breeder were to genetic skin test, then they would see if they found this, then this would change to 100% het, because they would have the analysis to show it. So guys, that's what I wanted to show you. So today we covered dominant and recessive. We covered incomplete dominant pretty much and recessive pretty much. Now the codominant, remember, there aren't that many of those. It's only one that I know of. So that's what you got to deal with, guys. And I hope this helps you to identify. I would encourage you to play around with this. And as always, if you like this, please like and subscribe. But more importantly, if you have any comments or suggestions, or if you have any just questions, just let us know here at Sphericreptiles and we'll get back to you. All right, guys? Hey, I hope you have a good day.